It is 3 o'clock, and I want to welcome everybody to the Environmental Engineering Graduate Seminar Series for the Spring 2021 semester. And today, I'm not only the seminar speaker, but I'm also the coordinator of the seminar series. So I just want to say a couple of quick words about that before I actually launch into the seminar. So what I hope to do this semester is really build on the very strong seminar series that Dr. Brian Barkdahl put together in the fall semester. And in particular, I hope to have a number of seminar speakers, not just from academia, but who are doing work related to environmental engineering and science in engineering practice, through work for government agencies or nonprofit organizations, so that our students and our faculty continue to get a big picture of our field, of the environmental engineering and science field. So I do still have a number of seminar dates that are open, have not been filled. So please do continue to send suggestions for seminar speakers to me. And if you have a topic that you want to speak about, please let me know that as well. In particular, the January 25th slot is still open. Also, I wanted to let you know that Angela Karanen is recording our seminars, and those will be posted to this Department of Civil and Environmental Engineering's YouTube channel. So if you want to go back and revisit a seminar or perhaps share it via social media, please do visit that departmental YouTube channel. And then lastly, for those who are interested in getting documentation of their participation in this seminar series to maintain their professional licensure, demonstrate continuing education, please reach out to Dr. Eric Seagren. He will be continuing to provide that information, that documentation. So with that, I'm going to switch gears now and go ahead and get my seminar loaded up. It's always a bit difficult to find a speaker for the first day of classes in the spring semester, so I thought I'd just go ahead and talk about some of the work that I've been doing on wastewater-based epidemiology. And so specifically, I'm going to be talking about the work that we've been done with this tool, wastewater-based epidemiology, to monitor the abundance of SARS-CoV-2. That's the virus that causes COVID-19. And specifically, we've been looking at this, working in this area on the Michigan Tech campus and throughout the Western UP communities. And my co-author on this presentation is Dr. Byung-Jun Kim, who's an assistant professor in mathematical sciences. And he's been starting to do some analysis of the data that we've been collecting on campus, and I'll touch briefly on that at the end of my presentation. What I want to do today is give you kind of an overall picture of this tool, this wastewater-based epidemiology tool, explain how it works and specifically is applied to monitoring SARS-CoV-2. Then I'll talk about some of the studies that I've been leading and the methods that we've used in those studies. And then finally, some of the results that we've obtained and what those results suggest. And feel free to ask questions if there are any. It can be technical or otherwise. Feel free to ask questions. I won't be monitoring the chat, but just speak up. So this tool, wastewater-based epidemiology, has actually been around for some time. And it can be used to track general trends in community health, aspects of community health that might be impacted by the use of legal as well as illicit drugs, alcohol consumption and tobacco use, exposure to environmental toxins, and infection with pathogens or disease-causing organisms. And historically, it's been shown that wastewater-based epidemiology can be used to identify outbreaks in poliovirus, hepatitis, track seasonal trends in influenza, and relevant to our current situation in my presentation today, track the spread of COVID-19 through communities. And it's really the current pandemic that has brought wastewater-based epidemiology to the forefront and highlighted the power of this tool. And basically, we can use wastewater to get information on community health, because when we consume drugs or exposed to toxins, our body's metabolism produces specific chemicals that aren't present if that exposure to a drug or toxin did not occur. And we call those biomarkers, and they end up in the urine generally or in the feces. And similarly, with many pathogens, when we're infected with them, we end up shedding those pathogens. In the case of COVID-19, we shed SARS-CoV-2 virus particles, and they end up in our feces. And so these biomarkers and these pathogens, or at least the genetic material left over from the pathogens, they find their way into the wastewater, and the signal associated with those biomolecules persists for some time. So that when we go and collect a sample of wastewater, and here over on the left, lower left, is a picture of Tom Wersiak up in Copper Harbor collecting a sample of wastewater from their treatment lagoons last summer. We can analyze that wastewater and look for the presence of those biomarkers or that genetic material, and get information on these different parameters, drug use and pathogen abundance within a targeted population. Let's look at some of the key steps based in wastewater-based epidemiology as it's applied to monitoring SARS-CoV-2. So up here in the upper left, we have what's supposed to represent a population of people. This could be the residents in a dormitory, in a nursing home, school children, or an entire community. The red people represent individuals who are infected or are COVID-19 positive. Again, when we're ill, when we're infected, we shed those virus particles. So the more people who are infected, the more virus particles will find their way into the sewer. And then if we can collect a sample from that sewer, and again, this could be targeted to a specific school, we might capture the flow just from a school or a residence hall or a nursing home, or we could collect our samples at the wastewater treatment plant and get samples that represent the whole community. So we collect that wastewater, which again still contains that virus particle. And this is a composite sampler, which collects samples at regular intervals. Then we can take it into the laboratory, and there we have to do a number of things. We need to concentrate the wastewater sample because the virus is quite dilute in the wastewater. And then we need to extract that genetic material out of the wastewater. In the case of SARS-CoV-2, that genetic material is RNA, ribonucleic acid. And then once we've extracted the RNA from the sample, we can quantify it and determine the total amount of virus that was present in the wastewater sample to begin with. So several aspects of this last process here, extracting and the RNA and the methods that are used to quantify or identify the genetic material are very similar or identical to what happens when you go to get a COVID test. So if you go to get a COVID test, they take a swab, take a human specimen, and then they extract the RNA. They don't have to concentrate it, but they have to pull out that genetic material, and then they have to determine if that there's any genetic material from SARS-CoV-2, from the virus that causes COVID-19. They don't really care how much virus is present. They just want to know if it's absent or present, if you're COVID-19 negative or positive. And what we're doing is different in that we don't just want to know that the virus is there. We want to know how much is there. We want to know how that virus abundance is changing over time, and spatially in some cases within a community. So let's look at how we can use these data then on virus abundance that are collected over time or perhaps over a geographical area. One of the most important ways in which we can use the data is to detect the emergence of an infection, COVID-19, within a community. So if we're sampling over time and we do not detect the virus, and suddenly we detect it in the wastewater and that the concentration increases, we know that we have an emerging infection within our community. And hopefully over some time, we see those virus levels decrease and provide evidence that we're beginning to decrease the number of infections or eliminate them from a community. And this can be used to make decisions about a number of things, but it may determine, for example, whether schools stay open or so forth. We can also use this information to potentially identify COVID-19 hotspots or potential exposure sites. So for example, if we're collecting samples initially at a wastewater treatment plant, again, we see the emergence of the virus in the wastewater at the plant. Now we can go up into the sewer shed and try to figure out where is that virus really coming from within the greater community. Is it associated with a nursing home, a school, a particular neighborhood? So we can go in and do some more refined testing from individual manholes and kind of narrow it down. And then that information in turn can be used to direct diagnostic testing. So if we identify, for example, that we're getting a lot of virus in the wastewater flowing out of a nursing home, public health officials may decide, boy, we better get in and do diagnostic COVID tests on all of the residents and staff in that nursing home. And also we can distribute other resources and do some targeted information distribution using this information. So just to kind of demonstrate that this approach works, that we can get information on the COVID-19 dynamics in communities using wastewater-based monitoring or epidemiology. I just highlight the results of two case studies. The first was one of the one here at the top of the slide was one of the first studies to demonstrate this idea. It was conducted early in spring of 2020 when we were just beginning to come to terms with the pandemic. It was led by Jordan Pescia at Yale University, so it was conducted in New Haven, Connecticut. And Dr. Pescia began monitoring the abundance of SARS-CoV-2 virus in what we call primary wastewater treatment solids. So one of the first steps in wastewater treatment involves using gravity to settle out the particles that are in the raw wastewater. And he anticipated that that would contain high concentrations of the virus, because we all know that the SARS-CoV-2 virus has those spike proteins that make it sticky, and we expect it to adhere to solid particles. So he began doing that work early in March, and then between March 19th and March 25th, he saw a large increase in the virus levels in those solids. So that's plotted in the lower plot. We have the total virus RNA in the solids. This is a log scale, and the date is on the x-axis. So he saw this increase, order of magnitude increase in virus levels. And several days later, the new cases reported in New Haven, this is on the top plot, and again, the log scale as a function of the date, began to increase as well. So the increase in SARS-CoV-2 levels in the wastewater preceded COVID-19 hospitalization and positive test results by several days, up to 6 days. And so that provided a lead time that the infection was beginning to emerge and surge within the community. Another well-publicized example of successful wastewater-based epidemiology was reported from by the University of Arizona in summer, early fall 2020. Their team began monitoring SARS-CoV-2 in dormitory wastewater flows beginning in July, and I should say that this is one of the largest, if not the largest, environmental microbiology department in the country. University of Arizona tested all residents for COVID-19 before they moved into their dorms in late August, and the wastewater surveillance team detected elevated SARS-CoV-2 levels in the wastewater from one of the residence halls, Likens Hall, in the week of August 23rd. They notified their administration, who then directed all of the residents of that dormitory to get another COVID test. And 2 out of the 311 residents were found to be positive for COVID-19. They were moved into isolation, and an outbreak was prevented in the dormitory. So that study really highlights one of the key advantages of wastewater-based monitoring. You might wonder, why not just test people? Why not just send people in for the COVID test? Well, the key is that we begin shedding the virus in most cases before we develop those symptoms of COVID-19. And that's really important because it's generally the emergence of symptoms that prompts people to go get a COVID test. So we should, as illustrated in this cartoon, see SARS-CoV-2 levels begin to increase before we get evidence of symptomatic COVID-19 cases. And this is also important when we have a community or members of a community who are resistant or unable to get a test. And it's also relatively cost effective because we're getting information on the infection rates or levels within an entire population rather than having to invest the money in testing each individual. So I really see these two tools, the COVID-19 diagnostic test and the wastewater surveillance or epidemiology as complementary assays. There are also, though, some real challenges associated with using wastewater-based monitoring to quantify SARS-CoV-2 or COVID-19 in a community. And one of the primary challenges is that the abundance of the virus is extremely low in the wastewater. So we're still getting information on just how much virus people shed when they're sick or even when they recover. But one estimate suggests that we have around 5,000 to 40 million copies of the SARS-CoV-2 genetic material per milliliter of feces. That's relatively high. But then that fecal matter gets diluted with all manner of gray water, the water from our showers, washing clothes and dishes and so forth. And so when we get to the wastewater treatment plant, it's thought that the amount of SARS-CoV-2 genetic material in the wastewater will range from 0.02 to 3,000 copies per milliliter of wastewater when we have active infections. Another challenge is the wastewater matrix itself. As you can imagine, wastewater contains a multitude of materials, and some of them inhibit the methods that we use to detect and quantify RNA. And those technologies are all based on something called polymerase chain reaction amplification, or PCR. So there are compounds in the wastewater matrix that inhibit that PCR reaction. So we have to try to account for that. And then in addition, wastewater contains a multitude of enzymes that work to chop up the genetic material. So it's a little bit of a race against time. If we want to detect and quantify this genetic material, we have to do so before the genetic material is destroyed. There's also some uncertainty regarding the best sampling methods. We saw in some of the earlier pictures, we can collect a grab sample, a discrete sample from wastewater at a specific point in time, or we can collect what we call a composite sample, where we have an automated sampler that removes a bit of wastewater at pre-programmed times. And different groups use different approaches, and that can make it difficult to compare the results from different studies. Another really important challenge is accounting for the amount of feces in a given sample that's collected from the wastewater. Recall that the virus that we shed is found in the feces. And that's important because if we look at as illustrated in this little cartoon over on the right, in the middle sample, the brown, we all know what that represents, the feces. We have two samples here this is supposed to represent two samples collected at different times, both of which contain the virus at the same concentration, represent the virus with the blue stars. So we see that if we have a larger amount of feces in our sample, even though the virus concentration within the feces is the same in the two samples, when we measure the second sample, it will register as an increase in total virus abundance relative to the first sample, just because we have more of that feces in the sample. And likewise, if we have two samples with the same amount of feces, but the concentration of virus within that feces increases, again, it will register as an increase in the absolute abundance of virus. And we need to be able to distinguish between those two phenomena. What we want to be able to do is normalize the amount of SARS-CoV-2 virus that we measure relative to the amount of feces in the sample. Another challenge is that we don't know how long people shed the virus. I mentioned this earlier. and the amount that people shed. It seems to vary somewhat depending on the individual. And this makes it difficult to relate the amount of virus in the wastewater to the total infection rate within a community. So this is another challenge that we have. So I'm going to switch gears now and talk about the studies that I've been involved with or leading, in which we've been doing wastewater-based monitoring of SARS-CoV-2. I first became interested in this technique and how we might apply it back in spring. And so I put together a small proposal with my colleague, Dr. Eric Seagren, and Dr. Ebenezer Tumban, who's no longer with Michigan Tech. And our goal was to understand, improve our understanding of what happens to the SARS-CoV-2 virus when it gets into the wastewater. We have quite a bit of evidence now that it's no longer infectious. It can't make people sick once it gets into that wastewater matrix. But we wanted to know if really demonstrate that and find out what happens to it during the course of the wastewater treatment process. And if we have solids, if we could just remind folks to mute your mic if you don't have an active question, that would be helpful. When we treat wastewater, we do produce materials called biosolids that are rich in organic matter and nutrients and have been treated to reduce pathogens. And so we try to beneficially reuse those, for example, by applying them to mining sites and so forth. So we want to also find out if the virus is present in those treated biosolids, and as well if they're applied to agricultural lands or mining sites, what happens to the virus if it's present at all in the soil and in any natural waters. So we obtained funding for that work from the Michigan Tech College of Engineering. I should say we obtained funding to purchase supplies, but not to fund students. So without students to analyze the samples, help me process and analyze them, not a lot happened last summer, but I did go around on a weekly basis and collect wastewater and wastewater treatment samples from our plant here in Houghton, as well as in Ironwood and Copper Harbor, and my colleagues at the Marquette wastewater treatment plant began collecting samples for me there. Then again, recognizing that we needed student help to move this work forward, and also seeing an opportunity to use this tool to manage COVID-19 and make decisions about COVID-19 on the Michigan Tech campus. We pitched an idea to the university administrators that we would collect wastewater samples, sample wastewater emanating from our different residential halls, and provide rapid feedback on SARS-CoV-2 levels in those wastewater flows. And my collaborator on that proposal, collaborators were Eric Seagren, again, and Karen Helt in chemical engineering. So the university was supportive of this idea. So this work is being funded by Michigan Tech's Office of the Vice President for Research. And here we collect wastewater from 4 individual residence halls 3 times a week, and from a combined residence hall flow 5 times per week. The investment of Michigan Tech into wastewater-based epidemiology in my lab, the capacity, led to my invitation to participate in a Michigan statewide pilot study. This has been a 3-month project that is just now wrapping up. It had really 2 key goals. One was to build the capacity or infrastructure for community health and research efforts throughout the state of Michigan, and also assess the potential for using this wastewater-based epidemiology to monitor COVID-19 dynamics in Michigan communities. The funding for this work was provided by the CARES Act, and was administered through Michigan's Department of Environment, Great Lakes, and Energy, or EGLE. The work was conducted by partnerships between testing labs like mine at Michigan Tech, local and regional health departments, and municipalities and treatment utilities. We were responsible for collecting and analyzing samples from the 5 western UP counties, so Baraga, Gogebic, Houghton, Keweenaw, and Ontonagon counties, and I'll talk more about that in a little bit. I want to take just a moment and talk about some of the technologies that are used to quantify SARS-CoV-2 and other organisms, as well as genes in all manner of samples. So it could be environmental samples, and these technologies are also used if you get some kind of genetic test. All are based on this PCR, polymerase chain reaction amplification. And basically, I don't want to belabor it, but there have been 3 generations of PCR. So when PCR first started, we had first generation PCR, in which we start with some genetic material. Again, it could be derived from wastewater or human specimen. And we subject that genetic material to some reactions with an important, what's really important is that we have specific primers, short sequences of DNA that will bind only to the target genetic material. And I'll talk more about what the target genetic material is in just a bit. And we then subject that mixture, the genetic material and primers, to a series of reactions in which the targeted genetic material is copied over and over and over. And eventually we get enough genetic material that we can do some analyses of that genetic material. But with this first generation PCR, we had no way of determining how much of that genetic material was present in the initial sample, be it wastewater, natural water, or a human specimen. So this was resolved with quantitative PCR, second generation. We still use those primers, those short DNA sequences that will only bind with our targeted genetic material. But we use those primers along with a probe that contains a fluorescent tag. And so when we make copies of that targeted DNA, we get more and more fluorescence. As we make the copies, the fluorescent signal increases, and eventually there's enough fluorescent signal that it can be detected by our instrument. And we keep track of how many times we have to repeat that reaction. We call those cycles, until we get a detectable amount of fluorescence. And then we can compare that cycle number, the number of times we have to copy the genetic material to get a detectable signal, to the number of cycles we need to get a detectable signal with standards. So before I could start this work, I had to obtain a standard, a stock solution of SARS-CoV-2 genetic material from a biotech company. And I obtained that, and I knew the concentration of genetic material in that stock. And then I make dilutions, and I subject each of those dilutions to this quantitative PCR process. And then we can measure the cycle number needed to get a detectable signal from each of those standards. And again, then by comparing that to the cycle number needed to get a signal for our sample, we can infer the amount of genetic material in our initial sample. So that's quantitative PCR, or second generation PCR. And then in the third generation PCR, or droplet digital PCR, we use a different approach. We don't have to prepare these standards, but instead we take our sample, so it might be a 5 microliter sample of my RNA or nucleic acid extracted from my wastewater, and we partition that into many, many droplets, at least 10,000 droplets. And those droplets are so small that each will generally contain 0 or 1 copy of the target genetic material. And then we subject each of those droplets to the PCR reaction, and again, over and over until we get a signal. And then we analyze each droplet for fluorescence. And so we can count the number of positive droplets relative to the total number of droplets, and again, determine how many copies of the genetic material were in the sample. So in some of the work that I'm going to be presenting or talking about, we use the quantitative PCR, and others the droplet digital PCR, or third generation. In terms of the target, what we're talking about is sequences of the RNA. So the SARS-CoV-2 virus is an RNA virus, and we've all are tired of seeing this picture, but the virus is represented here over on the left. At its core is again, the RNA, the genetic material that encodes all of the features of the virus, including the spike proteins, which are indicated with a S, some nucleoproteins, which are indicated with an N, the RNA genome, that's just the RNA sequence, encodes the membrane proteins, and also some, the envelope proteins and other features of the virus. So when you go get a COVID test in the United States, what they're going to be looking for when they say they're looking for the virus is actually the sequences of RNA or genetic material that encode for the nucleoprotein. They look for, quantify, or try to detect the N1 and N2 genes, we call them. If you get a COVID test in Europe, they would instead be looking for the RNA that encodes these envelope proteins. And there are other methods used in other parts of the world. But those are our targets. You'll see me refer to N1, N2, and E. In addition, recall that we really want to get a measure of the amount of feces in the sample. And to do that, we do something that sounds a little bit strange, but we measure the abundance of genetic material derived from the pepper mild mottle virus, or PMMOV. The pepper mild mottle virus is a plant pathogen. It infects peppers worldwide, and consequently, it's found in lots of processed foods, including hot sauces and curries. And so it's almost universally present in human fecal matter. And in fact, studies have shown that it's the most abundant virus in human feces. So if we can measure the amount of PMMOV, or pepper mild mottle virus, in our sample, that should provide a measure of fecal matter. And then we can normalize the amount of SARS-CoV-2 genetic material to the amount of pepper mild mottle virus genetic material, or fecal matter. Okay, so I'm going to switch gears now and talk about some of the data that we collected, have been collecting since spring at the Portage Lake Water and Sewer Authority plant located here in Houghton, Michigan, on the beautiful Keweenaw Waterway. So for those of you who are interested in these things, the plant is a relatively small plant designed to treat around 3 million gallons per day, actually treats closer to around 2 million gallons per day of wastewater these days. And this wastewater is generated in Houghton and Hancock, as well as some of the surrounding or nearby townships. It's a conventional activated sludge plant. For those of you who are interested in those things, we analyze the samples using the second generation PCR approach, quantitative PCR. The targets, SARS-CoV-2 virus targets were the N1 and N2 genes, and I also quantified the pepper mild mottle virus in these samples. Again, this work was inspired, or is being funded in part by the College of Engineering grant. So recall, one of the key goals is to find out what happens to the virus once it enters the wastewater treatment plant. So just a quick walkthrough of the wastewater treatment wastewater treatment process. Wastewater is pumped up using screw pumps just to get enough head in the water so that it can flow through the plant by gravity. And then we have some preliminary treatment processes to remove large materials, harmful materials that would harm the downstream equipment. So we collected samples at each point noted by an asterisk. So our first point is what we consider post-grit removal, and this we consider our influent wastewater. The first thing then that happens is that we use gravity to sediment out a large number of the particles. And again, our expectation is that a lot of the virus would be associated with the solids that settle out. So we collected a sample downstream of this gravity sedimentation step to check this hypothesis. And we also collected samples of the solids, and we still are collecting samples, I should say, of the solids that sediment out. Then we go to biological treatment and a second gravity sedimentation step. So we collected an effluent sample downstream of biological treatment in the second gravity sedimentation step, as well as the solids that settle out. The solids that are removed from the treatment process are subjected to anaerobic digestion to reduce pathogen levels and then dewatered. And the resulting product is considered a biosolids product, and we also collected samples of biosolids. So the red samples are the wastewater samples, and those are samples that I've begun to analyze in my lab. And so I'm going to be showing those results today. The blue samples have been collected but have not yet been analyzed. So those are the solids. Let's take a look now at some of the data that we have. So I'm going to walk you through these graphs. So in the upper plot, we have the cumulative COVID-19 cases reported in Houghton County by the Western UP Health Department. So recall that, or as we all know, Houghton County covers a large geographical area and has a relatively large population, around 37,000 individuals or so, I think. And only a fraction of those people actually deposit their feces into the Houghton Hancock sewer shed. So some of the cases here in Houghton County, again, that the feces that those people are producing will not be entering our wastewater treatment plant, but a fraction of it will. And also we can assume that some of the people who are diagnosed with COVID-19 recovered during this period and stopped shedding virus. But still, it gives us some measure of the amount of COVID-19 in our area. In the bottom graph, I have copied the absolute abundance of the N1 gene. This again is genetic material derived from the SARS-CoV-2 virus, but not normalized to the amount of fecal matter or PMMOV per mil of wastewater. We have three different colored columns here. One, the black represents the influent, the red represents the primary effluent. So this is the concentration of SARS-CoV-2 in the wastewater following that initial gravity sedimentation step. And then the green represents the concentrations following biological treatment in the second gravity settling step. So I'm not sure, my columns kind of spread out here and got a little bit wonky, but I have analyzed some samples from early June, early July, early and late August, and then more frequently samples collected during the fall. And over time, we're going to analyze some of the samples that are still frozen that we haven't gotten to yet. But we see an interesting picture. Early here in the summer, we had almost no cases of COVID-19 in our county, 1 or 2, and consequently, we did not detect the virus in our wastewater. We saw the first glimmer of a signal in early July, and then as cases began ticking up at the end of summer, early fall, we start to see increasing concentrations in the wastewater. So this really demonstrates again that when infections increase or emerge in a community, we can detect that signal in the wastewater. We also see some interesting results when we compare the virus abundance in the influent and in these other, further downstream in the treatment plant. Again, recall, I thought that we get a lot of virus associated with the solid particles, so I expected virus levels to decrease significantly from the influent to the primary effluent. In some cases, we saw that, but in other cases, the virus levels were actually, I should not say the virus levels, but the amount of genetic material derived from the virus was higher in the primary effluent than in the influent. And I have some ideas why that is, and I'll get back to that in a moment. And then we saw almost no virus at all or indication, any genetic material from the SARS-CoV-2 virus in the secondary effluent. I also should say that whenever we do analyses, we do them in triplicate. So we analyze 3 subsamples at a time. And for the influent and primary effluent, when we have a signal, it was present in all 3 of those subsamples, but for the secondary effluent, it was present in only 1 of the 3 assays, which further demonstrates that the concentrations were extremely low in the secondary effluent. When we normalize the amount of SARS-CoV-2 genetic material to the abundance of genetic material from that pepper-mild model virus, we see similar trends. So again, I've got cumulative COVID-19 cases over time in the entire county on the top plot. And again, now normalizing the SARS-CoV-2 genetic material to the abundance of PMMOV or fecal matter. We see again that the levels of genetic material from SARS-CoV-2 and the virus increase as cumulative cases increase. Here they're peaking at around December 1st, Whereas when we look at absolute abundance, it appeared to peak around December 17th. And in both cases, the amount of virus or genetic material from the virus appears to be on the decline. And this corresponds well to our total COVID cases in the county, which appeared to plateau somewhat in December. To try to get a little bit more information on this apparent decrease in the amount of genetic material from SARS-CoV-2 in the virus, I also compared those virus levels to the active cases. So beginning in early to mid-September, the county began, Western UP Health Department began reporting active cases that they were following. And again, this doesn't necessarily represent active cases within the Houghton-Hancock sewer shed, but within the total county. And here, what was most interesting is that when the SARS-CoV-2 levels began decreasing in December, that appears to correspond to a decrease in the number of active cases in our county. So in this bottom plot, I'm again reporting the total, just the absolute abundance of the SARS-CoV-2 genetic material per mil of wastewater. Getting back to the issue of why are we seeing relatively high levels in the primary effluent relative to the influent, we'll go back to our treatment scheme here. What is actually happening is the sketch is not complete here, but the way that the treatment plant is operating is they recycle their solids collected from the second sedimentation step back to the primary clarifier and then remove them through the bottom of the primary clarifier. In addition, after the solid particles are digested and squeezed in this filter press, the filtrate generated from this process is recycled to the primary clarifiers. So again, we do not believe that the virus is at all infectious. In fact, it's not intact in wastewater anymore. And so when we look for different targets derived from the virus, we might see perhaps the N1 target present, but not the E. And that provides evidence that the virus is being chopped up, but we are able to still, again, detect those biomarkers, those sequences of RNA in the wastewater. So I think it's these recycle flows containing chopped up bits of the virus genetic material, which result in the relatively high levels of the primary clarifier effluent. And we can validate that when we get to quantifying the samples of the solids themselves. So just to recap, we do seem to have a correlation between total and active COVID cases within Houghton County and the SARS-CoV-2 genetic material levels in our wastewater, and we expect that association to be stronger if we could just look at cases within the Houghton-Hancock sewer shed. And we have some additional work to do. We want to look at our RNA recoveries from the wastewater, and we want to, again, quantify the virus levels or genetic material levels in our solids and biosolids, and also quantify the N2 gene in our samples. And we're going to repeat this work for samples collected in Ironwood, Marquette, and Copper Harbor at their wastewater treatment plants. I want to switch gears now and talk a little bit about the work that we've been doing at Michigan Tech. Again, we've been collecting samples from 4 residence halls 3 times a week and from a combined residence hall flow 5 times a week. And here my colleague, Dr. Byung-Jun Kim did the work. He looked at our average concentrations in those triplicate assays and the standard deviations, and he compared those values to the number of positive COVID cases in residence hall divided by the total number of residents in those dormitories. And he looked specifically at data that was collected between August 28th and November 6th, and that really affects the findings because we weren't really able to generate results rapidly until sometime in October. So we had some big data gaps, and we'll talk about that more. And we use the same qPCR methods as we did for the Portage Lake Water and Sewer Authority plant. Just to give you a example of what the data look like, the green lines represent the ratio of positive cases in the dormitories to the total number of residents. And so those data are collected by Michigan Tech. And then the blue lines represent the ratio of that SARS-CoV-2 genetic material to the pepper-mild model virus genetic material. So again, we have lots of gaps here when we were getting our methods up and running, but then later, now we've been able to generate data on virtually a daily basis. So these data are kind of choppy. So Dr. Kim interpolated those data. And then what he was looking for is, are there similar patterns in our case data, the positive COVID cases, and the SARS-CoV-2 concentrations in the wastewater? So now the SARS-CoV-2 concentrations in wastewater are pink, and the case data are in blue. These are the smoothed out data. And he used those data to create what we call cross-correlation coefficient plots, and I'll try to walk you through those. Basically, when we have vertical lines that are positive, that means the patterns in the positive cases in our virus concentrations in the wastewater are following similar trends. So the amount of correlation between those two data sets is positive, excuse me. And then we see that on the x-axis, we're plotting lag. And when the vertical lines are in the negative territory, it means that we're getting information on the positive COVID tests before, or we're seeing positive COVID tests before we're seeing an increase in SARS-CoV-2 genetic material in the wastewater. And of course, we'd like to see things the other way around. We'd like to see evidence of the virus in the wastewater before we get those positive test results. And if that's the case, then the lag is positive. We say the wastewater data leads the testing data. So a negative lag means COVID cases are reported before SARS-CoV-2 genetic materials detected in the wastewater. So that was some data that were generated early on, but we did have examples of where we have detected the evidence of the virus in the wastewater before we have the positive cases. So this is an example where we detected a relatively high concentration of virus in the wastewater, and the COVID case data increased subsequently. And there have been other cases besides this one. So when we have that situation, then we get, positive correlation and also positive lag. So the SARS-CoV-2 genetic material in the wastewater leads or precedes the observed cases. Lastly, I want to wrap up by talking about the work that we've been doing as part of this Michigan statewide pilot study. There were approximately 20 teams, again, partnerships between testing labs like mine, health departments, and utilities and municipalities. The project was funded, it was $10 million of funding. We were team number 19. Again, our partners were the Western UP Health Departments and 18 utilities and municipalities. So the Western UP Health Department was extremely ambitious in collecting samples. I believe that we covered the largest geographical area and certainly processed one of the largest numbers of samples, despite the relatively small size of our team here at Michigan Tech. But so we got samples on the westernmost point from Bessemer and Ironwood. Southernmost, we went down to Lockview, Desert, and on the east over to Lance and Baraga, and on the north all the way up to Copper Harbor. So in addition to all of the other good work that they do for our community, we had our personnel from the Western UP Health Department driving and collecting, picking up the samples that were collected by the municipalities and utilities and then transferring them to me. So each day I would go receive the samples from the health department, and we have a chain of custody samples that had to be signed over for each set of samples. Since then, we've been processing the samples, about 70 per week. We've already concentrated the RNA in around 560 samples, and I took this picture of my biosafety cabinet one night when I was working late myself on the concentration step. It's extremely time consuming. So all samples have now been concentrated, and we're finishing up extraction of the RNA from those samples and analysis using the digital PCR technology, which we acquired as part of this grant. So again, we've built up our infrastructure and capacity for monitoring disease in our Western UP health departments. Our targets are the N1, N2, and E genes, which are derived from the SARS-CoV-2 virus. And we're also quantifying the abundance of another virus, which is called Feces, to basically quantify how much RNA we're able to recover from the samples and whether or not we're having inhibition of the PCR process. So I just want to wrap up by acknowledging the funding from the Michigan Tech's College of Engineering and Michigan Tech's Office of the Vice President for Research, which again built up the capacity for doing wastewater epidemiology in my lab and led to my participation in this statewide pilot study. Also, I greatly appreciate my partners at the wastewater treatment plants. I have a longstanding relationship with the Portage Lake Water and Sewer Authority and Zane McKenzie, the superintendent there, as well as Gogebic Iron Water Authority in Ironwood, Michigan. Dr. Seegren and I have been doing work with both of those treatment plants for many years now. Jeff Wasley is the superintendent there, and also the folks at the Marquette and Copper Harbor treatment plants. And also my dedicated band of students, John or JP Heron, Jess McQuinn, Alex Waypaw, Danny Cummins, Teresa Posse, Sam Arendar, and Bhavika Tarani have all helped with various facets. And especially I'm grateful to the team of students who goes out rain or shine to open up the manholes and collect the samples on the Michigan Tech campus. And I'd be happy to take any questions that you might have. This is just a shot of the freezer after collecting samples all summer. So we are full and trying to get this inventory whittled down. Are there questions?
Hi, Jennifer. This is really a fascinating topic. I'm just curious. You mentioned that when these like virus get into the wastewater system, they no longer make people sick. I'm just wondering, is that because the chemicals we add into the treatment process, or they just go naturally, like less problem?
Well, that's a great question. And I probably didn't, I had so many things I wanted to say and I might have overlooked that. So we don't know everything that happens. And this is an important topic, but there's probably a number of factors. One is certainly, as you mentioned, the treatment processes themselves. We have especially during the biological treatment phase, many positive, helpful microorganisms, bacteria, protozoa, rotifers, and things, which can graze on particles and inactivate them. It's not so much the extreme chemical or physical conditions which we encounter during water treatment. The only place we really encounter that is in the final step, which I did not highlight and I should have, and that is UV disinfection at our local treatment plant. So if there would be any stray genetic material, it would be inactivated by the UV light. But it's probably a combination of beneficial organisms and also just the enzymes themselves. So the wastewater matrix is challenging, and it really is probably to our advantage that the SARS-CoV-2 virus is an RNA virus, and compared to DNA, RNA is relatively unstable in the environment. So we all have our hands, and everything really that we touch is coated with, or has RNases, which are enzymes that actually cleave the RNA. So rather quickly, RNA can be chopped up. And so we have to actually go to some, pretty stringent measures in the lab to make sure these RNases, these enzymes, are not present in that processing, workflow, so that our RNA is not chopped up. But certainly wastewater is teeming with those enzymes. So it's really probably a combination of factors, and that's a great question. And there's a lot of work that we would like to do in this area, and not just the SARS-CoV-2 virus, but there are other viruses and pathogens as well that we want to understand how they're inactivated in engineered and natural treatment systems. And again, that was a major impetus for that first proposal. Are there other questions?
Yes, I did have a question actually. So I think it was the active cases graph that you were showing where there were, I think, 3 different spikes. And I believe the last one correlated pretty well to your data. Yeah, that one.
Yeah.
But could you explain why the data you collected doesn't really quite correlate with the first 2 spikes?
I can't say, but I can guess. So I think the biggest reason, well, I think there's probably 2 big reasons. One is that this first group of individuals may not have been really in the Houghton Hancock area primarily. So I don't have information on where these individuals are located and whether or not their feces is entering the sewer shed. So that's a major factor. The other is that we may need an accumulation of shedding individuals. So although we had maybe 200 or so infected individuals here in October, the number of people who are still shedding the virus in November or, down here in mid-October may have been higher. So again, because if we look at the cumulative number of people who are infected, we think that people shed for a number of weeks. And again, that makes it hard to correlate the amount of virus in the wastewater with the number of infected individuals. But if we're increasing the total number of infected individuals who are shedding virus, that will boost our signal at the wastewater treatment plant. So it's one of the pieces of this puzzle that we need to sort out is how long do people shed, and how to sort out that variability. But that's another great question. So I think it's the location and the duration of virus shedding that is at play there.
Okay, interesting. Thank you.
You're welcome. Any other questions? I could take probably 1 or 2 more additional questions.
I was wondering, this shedding phenomenon, are there other studies of other diseases that you could use to see what the pattern is?
Absolutely. Absolutely. So I think that again, it's the pandemic has really highlighted the power of this tool. And I think we're going to see it being used a lot more in the future. So if we get, information that we have an emerging illness, we can be on the leading edge. We can be prepared now once we can get information on what sequences within the genetic material we should look for, and we can look for those signals in our community wastewater flows. So it's been used historically to detect outbreaks of poliovirus, hepatitis, and look at patterns of influenza. Those are just a few. So really, there's been a few real pioneers of this wastewater-based epidemiology, primarily the group at Arizona State, led by Ralph Halden, but he's shown that it can be used for a multitude of things. But definitely there are many other viruses and pathogens that we shed in the feces. So yeah, wastewater definitely contains a multitude of pathogens, but this one is particularly interesting because generally we expect to see enteric pathogens in wastewater. So, waterborne, foodborne illnesses, we ingest them, and then it makes sense that they would be contained in the feces, which is what, the material that's emptied out of our intestines. But SARS-CoV-2 is a respiratory virus. We get infected through inhalation of the particles, and so it's interesting, and it's I think it's fortunate that it is still excreted, shed in the feces, despite it being a respiratory virus. But that, again, that is true of some other respiratory viruses like influenza.
So is there a pattern within the human body when you're, as you're recovering, you keep shedding and then it tapers off over time? Is that a correct possible explanation?
It is. It is. And again, we don't really know much about the shedding. So there's so much that, so much work that has had to happen in response to COVID-19. Obviously, development of treatment strategies and development of vaccines. And there's been a lot of work being done on the monitoring. And now, just now, people are starting to find the bandwidth to look at these shedding rates. So you have to be able to get samples from individuals over time, fecal samples, and, quantify those. And again, it's a laborious process. And we want to look at, a broad swath of our population. Shedding patterns are probably different for children compared to adults. And it might depend on how sick an individual is, how long they shed. So we're trying to figure out what those patterns are now and if we can make generalizations about them. So we're at 4 o'clock. I'm happy to take additional questions. I had my email up there on the last slide and my office phone number. So if you would like to chat about this more, drop me an email or give me a call. And also, as I mentioned, Angela Karanen will be posting this seminar to the Department of Civil and Environmental Engineering's YouTube channel. So thanks to everybody for attending, and we hope to see you again on January 25th. There will be no seminar next week for the holiday, but we'll be back on subsequent Mondays at 3 p.m. So thank you, everybody, and I hope everyone has a good semester.
Thanks. Good job.
Thank you.
Thank you.